Good afternoon, everybody. We are going to start the next section of our International Symposium on Biological Science. First of all, I'd like to thank Professor Andrea. Andrea, thank you very much for joining us during our International Symposium on Biological Sciences. Dr. Andrea Tarozzi is a professor of pharmacology and toxicology in the School of Pharmacy, Biotechnology, and Sports Science in Bologna University, Italy. I'd like to say that we are very happy you are here today. Professor Andrea has collaborated with our graduate program since 2017, and he also came to Brazil several times, participating of lectures, teaching our students in disciplines, and also collaborating in research with our colleagues. Dr. Andrea has been working with new strategies for the reduction of risk factors associated with acute and chronic neurodegenerative disease, as well as the identification of critical biological targets associated with the neuronal loss and neuroinflammation in neurodegenerative diseases during aging. Furthermore, he has studied neuroprotective drugs that would prevent the progression of degenerative disease. Professor Andrea, again, thank you very much for being so kind and for having accepted the invitation to be here. It is an honor having you here, so feel free to start your lecture. Thank you very much again.
Thank you, Manuel, for the presentation. I'm very happy to speak again with the Brazilian research, because I like to find a relationship or to maintain relationship. So now I start with my presentation. I hope that the quality of the image is good. So my presentation is the oxidative stress and nuclear NRF2R pathway in neurodegenerative disease. So I hope to focus some aspects that attract some interest about this topic, specific topic for neurodegenerative disease. So we need to start with some general consideration. So when we are speaking about neurodegenerative disease, we are speaking about a wide family of diseases that include Alzheimer's disease, Parkinson's disease, Huntington disease, amyotrophic lateral sclerosis disease, and the other kind of disease. All these neurodegenerative diseases are characterized for the chronic evolution, and the various symptoms. For example, the deficit cognitive for Alzheimer's disease, or some impairment for the movement for Parkinson's disease, reflect the specific neurodegenerative disease at specific brain region. So in the next slide, we can see, for example, that the involvement of a specific region and the neuron that justify specific symptom for the different neurodegenerative disease. For example, for Parkinson's disease, are involved the dopamine neuron, or for example, for Alzheimer's disease, cholinergic neuron that find the location in a pyramidal region and the hippocampus and the other region. So we start from this point of view that we have the selective neuronal deed in different brain regions. So it's important to know the mechanism that characterize this different neurodegenerative disease to find a new strategy to contain or to prevent this disease. About 5% of this disease are characterized by some specific genetic mutation, in particular for Parkinson's disease. For Parkinson's disease and amyotrophic lateral sclerosis, are two neurodegenerative diseases that find an important role for the specific gene. Also for Alzheimer's disease and for other kind of disease. In this table, you can see some examples, for example, genes that are involved in clearance of amyloid protein for Alzheimer's disease, or for example, for Parkinson's disease, we have several Perkin, for example, and the other kind of gene that are involved in neurodegeneration of dopamine neuron. However, this familial form represents only 5% of the cases, and therefore the vast majority of the cases are sporadic. It is known that environmental factor, lifestyle, other kind of factor, for example, vascular risk, can influence the appearance of this neurodegenerative disease, but still the major no risk factor for the most neurodegenerative disease is the aging. In this case, among the different neurodegenerative diseases, the Alzheimer's disease and the Parkinson's disease are two neurodegenerative diseases that are affected by aging. Particular Alzheimer's, Parkinson's disease, you can see that the strong increase of appearance of this risk and appearance of this disease in relationship with the age. It's a typical disease that appears in elderly individual. So, despite some specific alteration at the gene level for Alzheimer's disease, Parkinson's disease, and the other kind of neurodegenerative disease, now we know that we have several common neurodegenerative pathways in the different neurodegenerative diseases. So in this slide, we can resume the main common neurodegenerative pathway. For example, all the proteins that are involved in the clearance of misfolded protein that are involved in various neurodegenerative diseases. For example, amyloid protein for Alzheimer's disease or synuclein protein for Parkinson's disease. In parallel, we have other pathway that include the important impairment of mitochondrial function, because this aspect is very important, because all these diseases show a reduction of energy level, in particular the energy that from the mitochondria. The last system that is impaired is the system autophagic lysosomal pathway, that is very important, this system of a protein, because are involved together the protein folding ubiquity in proteasomal system about the clearance of a misfolding protein. So these two aspects is very important, as all the diseases share this aspect, in particular mitochondrial dysfunction and the impairment of the clearance of aberrant protein. We have another example that complete the last slide. That the consequence about the impairment of a clearance of this protein, not only but mitochondrial dysfunction. So these two aspects contribute in a significant manner so the neuronal function. In particular, you can show in the bottom of the slide about the synaptic toxicity and the network dysfunction. This aspect is very important because now we know that the neuroplasticity is very impairment from this different mechanism that we can find in similar manner in different neurodegenerative diseases. The last common neurodegenerative pathway is the inflammatory responses that involve in particular the microglia and the other kind of a glial cell. All these kind of cells that play an important role to trigger the different inflammatory process that can contribute to neurodegenerative disease. So, among the common neurodegenerative processes, we can include also the oxidative stress. The oxidative stress now we don't know if play an important role to trigger or to the progression of the disease. But in my opinion, the oxidative stress play an important role under different aspects, by direct and by indirect mechanism. In this slide, you can see a different mechanism that are involved the oxidative stress. In the center of the brain, you can see that the oxidative stress can contribute to neuronal death, to misfolding of protein, also mitochondrial dysfunction. This neurodegeneration is characterized together the neurodegeneration that involved the hematocephalic barrier that help to infiltration of lymphocyte, that lymphocyte orchestrated the action together the microglia to generate, to amplify the neuroinflammation. So, but we need to remember that for some specific neurodegenerative disease, we know some specific mechanism that underline oxidative stress. For example, for Alzheimer's disease, it is probably that amyloid protein play an important role to generate by redox reaction, to generate some radical species, including example peroxide hydrogen, and the other kind. So in this case, the deposition and the aggregation of amyloid protein can play an important role to generate a specific oxidative stress for this kind of disease. In similar manner, in Parkinson's disease, find a specific mechanism that contribute to oxidative stress. So we know that for Parkinson's disease, the impairment about the dopamine neuron function. Now we know that the dopamine, this neurotransmitter, play an important role to generate oxidative stress, in particular at intraneuronal level. In this slide, you can see that the dopamine, in presence of metal such as iron, can play an important role by function reaction to generate the different kind of reactive oxygen species and that evolve in oxidative stress. So Alzheimer's disease and Parkinson's disease show also some specific mechanisms that support the involvement of oxidative stress in neurodegeneration process. So, in this regard, it's very important to know all the mechanism, endogenous mechanism that play an important role to defend the neuron from the oxidative stress. We know that there is some master regulator of homeostatic situation at the neural cell. For example, it's very important that is a topic to today is the NRF2 is a nuclear factor 2. It's important factor that the nuclear factor that play an important role because control several genes that are involved in antioxidant defense of a neuron. But this protein NRF2 is ancestral protein because play an important role not only for antioxidativity, but also for a lifespan of our species. In this slide, you can see that there is a strictly correlation between the body size and the lifespan. In general, if increase the body size, increase the level of this kind of nuclear factor, and increase the lifespan of this species. So in the next slide, I think that it's very nice because in the table you can see this relationship also in the different mammalian species, from mouse, rat, pig, gerbil, and the other. So you can see that you have a strong relationship between the activity of this nuclear factor and the maximum lifespan potential. But we don't have a relationship, for example, for body weight. So it improves the concept that the activity of this nuclear factor is strictly correlated with the kind of mammalian species. So now we know we need to know the mechanism that I try to resume in some concept for the pathways that involve the nuclear factor 2 are involved another important protein that is KIP1. KIP1 crosstalk with nuclear factor 2 and control the activity of nuclear factor 2. So you can see that the KIP1 is a negative regulator of nuclear factor 2 and serves as a molecular switch to turn the antioxidant responses mediated by nuclear factor 2. In generally, in physiological condition, is very high the level of a KIP1. When we are in oxidative stress condition, in generally, the point one, the oxidative stress causes conformational change of a KIP1, in particular at specific cysteine residues in KIP1, that justify the next step, that is the release of a nuclear factor 2 from the complex together KIP1, so the nuclear factor 2 can enter into the nucleus and to active the transcription of several gene or nuclear factor 2 target gene. But this mechanism is controlled by other mechanism, because the level of a nuclear factor 2 at the nuclear level is controlled in parallel by the level of KIP1. So turning off of the signal KIP1 remove nuclear factor 2 from the ARE, that is the specific sequence of a gene, of a nucleotide that you find in DNA, that is very important because ARE is a downstream gene that under control of nuclear factor 2. In a cytosol, this complex is associated with the ubiquitin system that lead to degradation of nuclear factor 2. But from this two last slide, it's important to catch the important role of KIP1. It is the KIP1 that controls the activity of a nuclear factor 2. So the nuclear factor 2 control not only the gene expression of antioxidant gene, but control also the factors that are involved in neuroinflammation process, for example, including the nuclear factor KB, nuclear factor B. This kind of interaction find place in general in cytosolic level. Here there are some examples of the family of the gene that are under the control regulated by nuclear factor 2. We have a two important family of gene biotransformation detoxification and the antioxidant enzyme. I think that in this case are predominant than the other kind of gene. But very interesting because nuclear factor 2 control other kind of genes that play other kind of activity. For example, carbohydrate metabolism, lipid metabolism, including also the metabolism of iron. that justify that nuclear factor 2 is involved not only in neurodegenerative disease, but for example in diabetes, in cardiovascular disease, in this metabolic disease. Nuclear factor 2 is involved in several kinds of disease because are involved several different family of genes. So here there are other kind of a family of genes that are involved under the regulation by nuclear factor 2. So in this slide, find some data that record the expression, the effective expression of KEAP1 and the gene that control the expression of nuclear factor 2. So on the bottom of this slide, you can see that you find KEAP1 and nuclear factor 2 in both kind of cell, in neuron and in microglia. But in microglia, the level are higher than the neuron. So now we know that the nuclear factor 2 is a potential therapeutic target for neurodegenerative disease, in particular for Alzheimer and Parkinson's disease, where the oxidative stress play an important contribution to the evolution of this kind of disease. Here there is some example from postmortem tissue of a subject with Alzheimer's disease, and you can see that the presence of nuclear factor 2 at hippocampal neuron that confirm that nuclear factor 2 can a potential target for this kind of disease. This is for Alzheimer's disease. Also, in the same study, the author record also an important presence of nuclear factor 2 in not only neurons but also in astrocytes, so justify the involvement of this nuclear factor for the neuroinflammation that can characterize the Alzheimer's disease in hippocampus level. This is another example. So because I needed 5 minutes to finish my presentation, and on the basis of this evidence, of course, it's very important to find some molecule, some inducer of this nuclear factor 2. In this table, you see different kind of molecule that show the ability to increase the level of nuclear factor 2. Among, for example, all the wide family of polyphenol, for example, resveratrol, or for example, other kind is the, for example, polyene, protein factor, thicarbamate, isothiocyanate, different kind of molecules that with different mechanisms can active nuclear factor 2 and can potentially increase the antioxidant defense at the neuronal level. So the main pharmacological target in the complex of KEAP1 nuclear factor 2 is the KEAP1. because in KEAP1 you can find the different cysteine. Cysteine is a sensor cysteine that play an important role to release the nuclear factor 2 to able the movement of nuclear factor 2 versus the nucleus and to the transcription of several genes. So we have several residues that are sensitive to the action of different molecules. So very interesting is about sulforaphane, that is isothiocyanate, or dimethyl fumarate is another example. So in this picture you can see that the inducer can active nuclear factor 2 in two different manner, or through the modification of the redox cysteine, or by the phosphorylation of a nuclear factor 2. In general, the inducer work on cysteine at the KEAP1 level. In both cases, we have the release of a nuclear factor 2 that can enter to the nucleus and to activate several genes for detoxification, antioxidant, anti-inflammatory activity. So, in 2 minutes, I wanted to see that an example about sulforaphane metabolite, in particular sulforaphane and leucine, that we can find respectively in broccoli and rocket salad, is two important vegetables that can contribute to the release of these two kind of phytochemical that show a strong ability to induce the activation of nuclear factor 2. But it's very interesting because sulforaphane and leucine is in bidirectional metabolism, means that if I take leucine, I can develop a sulforaphane in my body. By contrast, sulforaphane conversion in leucine in my body. So it's very, in general, the leucine is metabolite of sulforaphane. So in this case, it's very important to consider if we can find this molecule at tissue level. In this slide, you can find that the brain where the author find the high level of sulforaphane that justify the use of this kind of phytochemical for neurodegenerative disease. In similar manner, the different metabolite of sulforaphane found also in the brain. It's very interesting because now we know that the sulforaphane together the metabolite can place in the brain also the different tissue. In this experiment, we saw that the ability of both sulforaphane metabolite to active the activity of the nuclear factor 2, and this increase of nuclear factor 2 justify the increase of antioxidant molecule, including glutathione, the decrease of redox status that justify the increase of glutathione, and also the ability to correct the status redox that contribute to neuroprotective effect in terms of reduction of neuronal death induced by neurotoxin specific for Parkinson. We confirmed this activity against the hydroxypropamine in vivo model of Parkinson's disease by injection of neurotoxin in the brain at basal ganglia basal, and we record the similar effect. We have the activation of neural factor 2. We have the increase of glutathione, and we have the decrease of neuronal dead in similar manner in vivo model. So in this picture you find some immunohistochemical sections that record the ability of a sulforaphane to counteract the neuronal death in terms of DNA fragmentation and in tyrosine hydroxylase positive cell, in positive cell. So also similar activity for glutathione and neural factor 2 in Parkinson, in vivo Parkinson model. So I think that my goal is to do some keynote that play in interest for some research. Thank you for your attention. Many thanks.
Thank you, Professor Andrea, for this beautiful presentation. Very, very interesting. So let me take a look at our chat. We have one question. Ericles Mesquita said, "Hello, Andrea. Congratulations and thanks to the lecture." And he says, "I'd like to ask if regular stress, such as physical exercise and long-time fasting, can activate NRF2 ARE pathways."
Hi, Ericles. A good question. So we know that the physical exercise, physical activity is very important because increase the activity of this pathway. So we know that the physical activity find a positive relationship between the healthy for the brain. Under the different mechanism, there is also the potential of this kind of pathway. It's very sensitive, not only for oxidative stress, but for other conditions that can induce some stress, not only reactive oxidative species, but other kind of species stress.
Okay, Andrea, we have some compliments from Professor Paulo César Ghedini, from Jessica Souza, Luana Manso. Okay, thank you, guys. I think we have another question from Ericles. How specific we can produce a new drug with the NRF2 as the therapeutic target without changing other pathway or induce side effects? Drugs with this action is already used as treatment for NDDS. NDDS, it is a question. Sorry.
Okay. I start from the last question. So now there is a drug for multiple sclerosis. Is the dimethyl fumarate. Dimethyl fumarate show anti-inflammatory, in particular anti-inflammatory activity, through its ability to activate this pathway. But this question is very important because we know this aspect from 10 years ago, but now we have a few molecules that play an important role for therapy. Why? Need to pay attention because now we know that the activation of nuclear factor 2 can associated to some side effect, in particular for cancer, because the question open now is easy to understand because because we know that several cancer is associated to high level of nuclear factor 2. So it's not too easy to discriminate this activity, but is a question open. It's a question open. You find several papers about this aspect in the last year.
Thank you, Andrea. Unfortunately, we don't have more time for questions, but Andrea, if you agree, we can put your email contact using our YouTube channel, okay? So, in the name of the organizing committee, I would like to thank you very much for having participated of our meeting, Andrea, and I hope you have also enjoyed this moment with us. Thank you very much. So now we are going to... Thank you, Andrea. Thank you very much. Now we are going to continue with our next presentation. And for that, I'd like to call Professor Mauro, who is going to introduce Dr. Friederike Klempin from Germany. Dr. Friederike, thank you very much for being here. It's an honor having you here. Mauro, it's up to you. Thank you.
Thank you, Mauro. Hello, Rika.
Hello, boa tarde.
Boa tarde. Hello, everybody. It's nice have you here today, and have this chance to introduce my colleague and a very intelligent researcher, Friederike Klempin. Friederike is a biologist. She studied at Humboldt University in Berlin. And she did she got a neuroscience degree on Humboldt University and Max Delbrück Center in Berlin. She had the chance to be a senior scientist here in Brazil, in the Federal University of Minas Gerais, and she is now a researcher, PI in University Medicine Berlin, and she is one of the she is a very important scientist working with neurogenesis and the whole of serotoninergic neurons in the brain. So it's our pleasure to have you here today, Rika. Please, we would love to see your presentation and talk a little bit more about your new results.
Boa tarde para todos. I hope that it works. And thanks, Mauro, for the nice introduction. Can you hear me? Can you see the presentation also?
Yes.
Okay, perfect. So, thank you. I'm happy to be in Brazil, at least partially online. And I will give an overview of my work on neuroplasticity and, as Mauro already said, of course, on serotonin and the hippocampus and a lot more, and also show you some recent data. First, I give a short introduction of what is neuroplasticity, what is adult neogenesis. So neuroplasticity enables the adult brain over a specific species. For sure, we study rodents and the human brain. So neuroplasticity enables to adapt to environmental changes and respond to novel experience by rewiring upon learning. Thereby, changes occur in synaptic structure, strengthening or weakening synaptic connections, but also, excitingly, the generation of new neurons. And it also changes occur upon that in dendritic spine, membrane properties, or DNA regulations, but also, and I will come to that, in neurotransmitter release. such as serotonin and on microglia activity. So without neurogenesis, I will have to choose my pointer. So comprises neural stem cells that persist in specific niches that give rise to amplified progenitor cells that become neurons. We can detect that, and this happens throughout life. We can detect that by injection of BRDU, thymidine analog, which gets incorporated during S phase of cell proliferation and can be stained immunohistochemistry-wise. So through a variety of species, as I already mentioned before, stem cell niches persist, which give rise to neurons, first detected in rodents by Altman and Das almost 60 years ago, which is interesting and exciting at the same time. And what you can see here in this graphic is that the smaller the animal, the bigger is the neurostem cell niche and cells migrate throughout the brain. However, with brain size, the number of stem cell niches and neurons decrease. So it's hardly detected in humans, but it does occur. So back to the rodent model. So everything which you see here in white is a non-neurogenic region. In green, we have two established neurogenic niches. One is the subventricular zone olfactory bulb system, where new neurons are continuously generated, contributing to olfactory information. So they can adapt in the environment to novel experiences. The other region, which I study, and lots of other researchers, is the subgranular zone in the dentate gyrus of the hippocampus, involved in learning and memory, and I will go in detail a bit later. Recently, a third region has been discovered, the hypothalamus, here specifically the third ventricle. However, also it may be a stem cell niche, it may not, and a highly plastic region, it may not be coming to newborn neurons. So I did, during my career, focused on the dentate gyrus as stem cell niche and characterized in detail progenitor cells, for example, detected by SOX2 expression, then an intermediate phase where double cortin is the characteristic marker. So these cells which express double cortin for sure become neurons, then characterized by NuN. So they change their direction and migrate within the small layer of the granular cells. In comparison to the non-neurogenic region, for example, the piriform cortex, which is approximately here, they are also double cortin positive cells. However, they are strictly post-mitotic. So they are not plastic as they are in the neurogenic niche of the hippocampus, which can be also characterized by electrophysiological properties. And I also studied, which I show another data in the next few slides, the hypothalamus, and we detected also stem cells persist, which are called tanycytes, and they do express an early lineage marker SOX2. They're not co-labeled with NuN, so they do not become any neurons. Okay, the hippocampus, one of the three stem cell / neurogenic regions. The hippocampus is involved in learning and memory. This holds true for mouse or rodents and humans. It is involved in learning and memory processes, in emotion. It can distinguish pattern separation and is highly involved in synaptic plasticity. So neurostem cells persist and they respond to both intrinsic signaling and external stimuli, which both promote a neuronal fate of these cells. My research focuses on serotonin, the important neurotransmitter in the adult brain, but also the neurotrophic signaling brain-derived neurotrophic factor. The prominent work by Henriette van Praag, you may know, shows that running, so voluntary real running in rodents, strongly enhances the reservoir of stem progenitor cells, which then can be used and get functionally integrated into a circuitry. A negative stimuli of neurogenesis and brain function and neuroplasticity occurs in injury or pathology, which we just learned before by the talk of Andrea Tarozzi, for example, in Alzheimer's disease. Okay, during my career, I was fascinated by serotonin, the neurotransmitter, which is the most widespread monoamine in the entire brain. It is pro-neogenic, so it supports the generation of new neurons, and it is also involved in antidepressant therapy. Serotonergic neurons are located in the brainstem raphe nuclei, and fibers terminate throughout the system, mainly the limbic system, where they synapse with neurons and interneurons of the dendrite gyrus. They exert their effects through a variety of serotonin receptors. It's approximately 15 now, which are mostly G protein coupled receptors. So I went on to study by targeting the serotonin system to enhance neurogenesis, because we do want neurons to be born in the adult brain. And I did it pharmacologically wise by, for example, targeting the 5-HT1A receptor with an agonist that boosts the number of proliferating cells, while the antagonist suppresses the number of newborn neurons. The opposite holds true for 5-HT2C receptor, so I established an opposed action of different serotonin receptors within the lineage of a neuronal fate decision. So negative serotonin signaling is a hallmark of antidepressant or of depression and can be used as target for antidepressant therapy. And here, state of the art is the use of a selective serotonin reuptake inhibitor, which inhibit the uptake of serotonin, so increases the availability of serotonin in the synaptic cleft. One drug is called Prozac or fluoxetine, and giving it for 3 weeks increases the number of newborn neurons in the dendrite gyrus and leads to increased mood in patients. Back to the other region, the stem cell niche of the hypothalamus, and here I studied the effect of serotonin together with a master student in food intake and metabolism. And we made use of mouse that lack brain serotonin. I will go into detail in the next slide. And we studied what happened at the stem cell niche. And you can see here, all the black dots are newly born cells stained with BLU. And we see that there's an age-dependent effect on these cell numbers, while at P42, so 6 weeks after mouse were born, we have an increase in the number of BLU positive cells. It declines by aging, and at 1 year, you have only stem cells left at the third ventricle. However, there's a decrease in the absence of serotonin. Another finding is even more interesting, that after feeding high cholesterol diet for 6 weeks, mice get fatter, which is normal, they gain weight. However, our TPH2 knockout mouse, so the mice which lacks brain serotonin, did not increase weight. So we do contribute with that result to the common knowledge that serotonin is not only involved in the brain's neurogenic niche and depression, but also in energy metabolism and contributes to satiety and food intake. So making use of this model, depleted of brain serotonin called tryptophan hydroxylase 2 deficient mice. This is the main enzyme for serotonin synthesis. I went on to study the effect of serotonin in the hippocampus and use the Henriette van Praag model of physical exercise. So we had 2 groups, mice at baseline, so they were held in normal cages for 5 days, and mice that had access to voluntary wheel, and they are really running overnight. They run 12 hours non-stop overnight. So we were surprised to see first that there's no difference at baseline numbers of proliferating cells, giving the significant studies supporting serotonin is a neurogenic factor. So you would assume no serotonin, you will have no proliferation in the hippocampus. That's not true, and there may be a compensatory mechanism. However, and another surprising and exciting result, we found that the running-induced increase, so running really boosts the number of proliferating cells, was abolished in mice that lack brain serotonin. So we established that actually serotonin is the link on running-induced increase in cell proliferation in the dentate gyrus. So we went on and looked at cell level and found that there's a decrease in our stem cell population that was accompanied by increase in caspase-3 expression, which is a cell death or a apoptosis marker. And also we found that after running, which already increases the number of microglia cells, it is even more enhanced in mice that lack brain serotonin. So the activated EVA1 positive microglia cells are strongly enhanced in lack of serotonin. Further studies show, as the studies we did, show that BDNF, the brain-derived neurotrophic factor, is also much increased or highly increased in the brain of serotonin deficient mice, maybe being the compensatory factor to keep baseline cell proliferation. And the exciting link between serotonin, BDNF, and microglia, which do release BDNF, will be of further studies, and I will show you preliminary results shortly. First of all, or back to the serotonin depression link, we here studied the interplay of the generation of new neurons, serotonin, which is a pro-neurogenic factor and used as antidepressants, and BDNF signaling. Since only a few or a quarter of patients do actually respond to common treatment, it is important to study basic research on that topic. What we did was we treated mice with electroconvulsive seizures, which is still a therapy used in humans who suffer from major depression. So we did a seizure session of 5 days, and after 3 weeks, measured adult neurogenesis and BDNF protein signaling. What we found was the known increase in adult neurogenesis and in BDNF protein levels that are hallmarks of electroconvulsive seizure therapy is diminished or lack in the absence of brain serotonin. So we do show that serotonin is and contributed to the effects of this therapy, and also found a correlation of BDNF and BRDU levels, which is positively correlated in wild type, but abolished in the lack of brain serotonin. So coming to the new part, preliminary data part, microglia, serotonin, and brain-derived neurotrophic factor. We had recently published a review together in the special issue, which we edited together with Mauro, on microglia function in the hippocampus. So microglia are the resident immune cells of the brain, and they become activated in response to negative stimuli, which could be pathogen-related or damage-related. And they do activate the serotonin of cytokines, which can be pro or anti-inflammatory, but in the first reaction, of course, they are anti-inflammatory to keep the maintenance of, to keep homeostasis of the brain. And neurodegenerative diseases, such as Alzheimer or depression and others, there's a microglia dysfunction of providing the first immune response. This image should not be too confusing, but it shows that there's also already an interaction of microglia cells at baseline. so in a normal homeostatic environment without any inflammation. And microglia can be found in close proximity to precursor cells of the dentate gyrus and to mature neurons of the dentate gyrus, which express the BDNF receptor TrkB. In their role being in the hippocampus at normal baseline levels, they take part in neuroplasticity. They control the neuronal cell pool by phagocytosis of cells which are apoptotic or they are not needed anymore. They regulate synaptic plasticity via the release of TNF-alpha, a pro-inflammatory factor, and of BDNF, an anti-inflammatory factor. And they do respond to exercise, which I had shown you earlier. They also respond to serotonin signaling. So synapses, fibers terminate the dentate gyrus, as I had shown before, and do release lots of serotonin in the dentate gyrus niche. So interestingly, recently serotonin receptors have been discovered or detected on microglia cells, responding to low or increased levels of serotonin release, which is all exciting and can be used and studied now further to understand the link of microglia inflammation and neurogenesis. And to emphasize some factors, one is the 5-HT2B receptor, which we focused on in some data I show next, which can either go the way and it's a G protein coupled factor and can activate phospholipid cascade, which then leads to calcium signaling release and EBA1 that is important to phagocytose and remove apoptotic cell, which the microglia cell does at the normal stage. It can go the other way via the cyclic AMP signaling and block the release of pro-inflammatory factors while enhancing the anti-inflammatory factors such as BDNF. So how does it work? And we studied this using using rats in this case, which also do lack brain serotonin, the tryptophan hydroxylase 2 deficient mice. What we did, and that's a study together with my PhD student from Kaliningrad, Andrea, we fact sorted microglia cells and then did PCR or cell culture or calcium imaging to identify difference in macro expression, in mRNA expression between wild type and knockout rats to see the effect of serotonin on microglia and so on. So what you can see here, first result we received was that actually doing fact sorting of cells, we found that the knockout rats had less cells while sorting cells from hippocampus, yet the proportion of CD11B microglia cells was increased. That was one first interesting result shown here in the quantity. So the hippocampus has more microglia cells out of single cells counted. And we already checked and thought then maybe other cells are increased, and we checked for astrocytes, yet we could not detect an increase in, for example, astrocytes. So we went on to study the inflammatory and pro and anti-inflammatory factors, and we found that BDNF is increased in two regions. We checked the hippocampus as an important region, then the cortex, which we always use as control region, the prefrontal cortex. However, it turns out now the prefrontal cortex may actually highly involved in the effects of serotonin on hippocampal function. So we found that the anti-inflammatory factor BDNF is increased in female and male cortex of TPH2 knockout rats. We looked at the 5-HT2B receptor as the already known factor expressed in on microglia cells, and we found an increase in cortex of female and male TPH2 knockout rats, so it was not significant or even decreased for the hippocampus. So that already shows the 2B receptor may be playing an important role in the cortex. We looked the opposite factor, the pro-inflammatory factor TNF-alpha, and we saw that while anti-inflammatory factors are increased, pro-inflammatory factor is decreased, at least in female cortex, while IL-6, interleukin-6, is also increased. So these two factors are in opposite direction, and we have to have further data on that. So then we were also interested in what other serotonin receptors are expressed on microglia, and we discovered a battery of experiments, male versus female, different ages, P8, P21, postnatal day, P56, then cortex versus hippocampus, and we found that at early age, the 5-HT1B, 5B, 2B receptors expressed, the 2B receptor expressed at all ages, while the 5B receptors downregulated, and with the 5A receptor being upregulated. So there's an alternating expression of receptors depending on age in wild type. If we go into more detail by qPCR, here shown for the 5-HT1B receptor, we can see in control that indeed the 1B receptor goes down, age dependent in male and female of cortex and hippocampus, while in the knockout rats, it also goes down, but maybe coming up a little for male and female knockout rats at P56, which is an exciting result. We have to go also here into further detail, and clearly showing the absence of 5-HT 5B receptor at older age at P56. So these are preliminary results, and we continue these studies. So the 5-HT2B mRNA is strongly expressed by microglia cells with a significant increase in cortex. Now we also want to see, of course, the protein and the receptor expression in vivo in the hippocampus. And we were surprised to find, we couldn't find any expression, and there's also rarely markers to show serotonin receptors, but there is one for 2B. And it is nicely expressed in the dentate gyrus. However, it is expressed on neurons and not on microglia cells. Here shown in red, and here shown in a bit higher magnification in green, so you can hopefully see the tiny little dots on neurons and in the subgranular zone, so the neurogenic region of the brain, while it is even seemed to be further higher expressed in the serotonin knockout mouse. So I think I was a bit fast. That's already the summary of my talk. So there are neurons that are continuously generated in specific and small restricted regions in the adult brain. The so-called adult neurogenesis in hippocampus is, for us, the most important one, and is involved in running-induced changes in neuroplasticity, brain plasticity. And my data, our data, show that serotonin is the mechanism that mediates the running-induced changes in plasticity. Then we can see clearly a link between serotonin and BDNF interplay, both in depression, but also in microglia interaction, that do answer or correspond with serotonin. They do release of BDNF, which then in turn affects neurogenesis and neuroinformation and neuro and transmitter signaling transmission within the functional network of the hippocampus. And some pictures. This is Berlin, and it's even as dark as it's on the picture. Then studies were done in the BADA research team at the MDC before. This is the master student Marike, who I did together the hypothalamus story, and then my students and colleagues from Kaliningrad. And I thank you for being allowed to be here to have this presentation. And I hope I know how to get out of this. Hello. I cannot hear or see anything.
Hello? Are you hearing me?
Now I can hear you.
Okay. Hi, Hika. Thank you very much for this very interesting presentation. It's very nice have someone here talking about neurogenesis and neuroplasticity. I have some questions here, and we have also several ones in the chat. I will start with the questions in the chat, and then we can talk a little bit more about other hot points here after this. So here, João Batista, he thanks for the great presentation, and he asked if your lab it's now working on the effect of serotoninergic psychedelics like psilocybin, DMT, or LSD in these models of neurogenesis.
Yeah, psilocybin. No, I do give lectures and I read all the papers about it, which does affect the 5-HT2 receptor subtypes particularly. But no, we did not do it, and I doubt that we will get an animal allowance to do that here. I'm not sure. Yet it will be interesting to see because like psilocybin does have an opposite effect, and also on the amygdala, which I did not talk about it today. So actually, it's a good idea to do. So maybe we can have some collaboration with Brazil.
That would be very interesting to try, right?
Yeah.
He also are asking about the use of these drugs as alternative therapeutic drug for traditional ones, probably for depression or anxiety treatment. What do you think?
So I think so. So for psilocybin, it's also a hallucinogenic drug. So I'm not sure yet whether it's allowed to do it, but I believe in small doses it shouldn't be too harmful. Besides, now in Berlin, we're gonna legalize cannabis for sure. And the second I wanted to say, ketamine now it's in high use, which we also actually do not know how it functions. So it acts very fast. So it cannot function via Neogenesis, for example, because therefore it needs 3 weeks. So there's a few drugs now which have a fast effect, but we should need to study what happens in the brain and maybe have long-term consequences, which we don't know yet about it.
Yeah, that's very interesting. These days, people are working on these new drugs that increase BDNF, right, very fast. But we still need more information about it, right?
Sure, there was this recent paper in Cell which shows that the ketamine right away targets on track B on neurons, track B signaling. So,
well, Professor Carlos, he have a very interesting question. It's also one of the points that I wrote here. Well, we know that there is a relationship between neuroinflammation and depression, right? And you show here that microglia it's important in the serotoninergic pathway, right, in hippocampus. So he asking about if serotonin it's able to modulate glial cells astrocyte function and inflammatory status. And he was wondering about the whole of the blood-brain barrier in this scenario.
So, two things I have to mention. First of all, now for the coronavirus studies or clinical trials or in clinic, they have used fluoxetine, for example, which shows both. So it does increase mood and it does decrease inflammation. So giving antidepressants early on in inflammation, it's actually a good, or maybe a good strategy to improve from both inflammation and depression. Second of all, the blood-brain barrier. So the problem with serotonin and the blood-brain barrier, you have two independent systems. You have the peripheral system of serotonin and you have the brain serotonin, this TPH2, which I forgot to mention during the talk. So they are separate systems. So serotonin does not cross the blood-brain barrier. So you need to have you can do something in the periphery, which maybe then in a secondary effect affects the brain, or you target right away the brain. Now,
did you have the chance to look the microglia activation and see some cytokines related to neuroinflammation, or even the metalloproteinase that will be released by microglia in your studies, in your lab?
So the release of cytokines now on microglia, we do its current study. So I showed the decrease in TNF alpha and increase in IL-6, for example, in microglia cells in cortex, not hippocampus. And we continue doing it. So it really takes a lot if you only have a 96 well plate to study all the as we have so many samples to study at one time, and in Kaliningrad they have 96 well plates, while we here have the 348 well plates, which you can have a faster qPCR. But yeah, we will study this, but we already found the opposite results for TNF alpha and IL-6, which is kind of interesting, and probably at the end we have so many results we cannot link to each other. So, but let's see.
That's very interesting.
And we also have a knockout mouse for peripheral serotonin and a double knockout. So maybe we could combine things there and see.
Yeah. He also asked about the whole of serotonin in the vascular, neurovascular vessels, right? But the point is, can you relate serotonin, the regulation of neurovascular contact, and the inflammation of the system?
I think no. So with serotonin, the good and the bad thing is it is released via bouton en passant. So it's continuously released on spines along the axon. So it's difficult to distinguish whether it's clearly at the synapse, whether it's extracellular, whether it's intracellular. So we try to do it via receptors.
Well, I have
maybe
a question.
With mass spec. Yeah.
I have a question from Professor Daniel. He asked, well, he said that sleep deprivation reduces neurogenesis. So the question is, could serotonin also be involved in this effect? There is a lot of evidence suggesting that cholinergic neurons are involved. Could you comment about it? This relationship between cholinergic and serotoninergic neurotransmissions in hippocampus?
So I in general, no, or in more specific, I cannot. In general, I know that, for example, in the mice which are depleted of brain serotonin, we also have all other transmitters are reduced, but only to a certain degree, so up to 70%. As 30% reduced, so they still have like 70% left. And however, at night, serotonin is translated, metabolized to melatonin. And melatonin does not occur in C57 black 6 mice, for example, which is always our background in mice. So we could not study. So we would have to give melatonin first and then study serotonin effect. Let me read the question again. Yeah, so we do know, as we don't know whether our mice are sleep deprived, but we know the opposite. They are highly active. So no serotonin actually increases their activity, but they do not run more, for example, or run less. So their activity is pretty normal. Yeah.
Well, we have a last question from Bruno. He asked about the...
Hi, Bruno.
Hey, Bruno. He asked about the physical exercise. Actually, I had a question about that. So the point is, we know that different types of exercise can induce different levels of neurogenesis, right? Could you talk a little bit about these differences? And the question is, if you know about the relationship of serotonin, BDNF, and memory in mice that you are working on right now.
So the BDNF and running, running exercises. So yeah, so you have the treadmill running used in studies, which is called the forced exercise. So people are already not happy. Mice are already not happy to being forced doing this exercise. So there's a co-factor that may not lead to increased neurogenesis, because stress may decrease neurogenesis. So that's why we use voluntary wheel running. So the mice run or not, but we make sure they do run, and they do actually run. They love to run. We have other models for bradykinin or the angiotensin enzyme 2, which we can monitor. They do run less, so then we have to counterbalance the effect on neurogenesis. But if you run 3 hours per night as a mouse, or as a human being probably, you boost your pool of cells that can be recruited to become neurons. So we did memory tests. I did not measure BDNF levels. So BDNF is also increased after running, for sure. I'm reading the question in between. However, BDNF, you can only measure, so it's shortly produced. So you can only see a super increase like half an hour after running. It will not last. So you cannot overexpress it. Well, yeah.
No, that's interesting.
And
so,
memory.
Yeah,
we did memory tests, but.
Hika, thank you very much for your time. Your presentation was awesome.
Yeah, thank you.
Yeah, and it's always a pleasure having you here. We will now we'll start the next presentation. But if someone here in the university, in our program, if you have some questions, specific questions for Friederike Klempin, please let us know. I will put the email of the professor here in the chat, and then you can send direct for her. Okay? So Hika, thank you very much. See you later. Bye.
Bye.
Hey, Manuel. Hey, Mauro. Thank you, Mauro. Thank you, Dr. Friederike, for this beautiful presentation. And I promise you, I will try to pronounce it, to practice your name in order to pronounce it correctly. So let's go to our next activity, our next presentation, who is going to be delivered by Rinaldo Rodrigues dos Passos Jr. Good afternoon, Rinaldo. First of all, I also would like to thank you very much for being here, participating in our International Symposium on Biological Sciences. Rinaldo Rodrigues dos Passos Jr. has a degree in pharmacy from the Federal University of Mato Grosso, 2018, a master's degree in basic and applied immunology and parasitology from the Federal University of Mato Grosso, 2020, and currently is a PhD student in biological sciences from the Federal University of Goiás in the Laboratory of Vascular Biology and Histopathology. In fact, I have to thank Rinaldo because he's a member of our organizing committee and has been working a lot in order to organize this meeting. Thank you so much, Rinaldo, for the support. So feel free to start your lecture. Thank you very much again.
Thank you very much, Professor Manuel. Can you see my presentation?
Yes, Rinaldo, everything is okay.
Can you hear me?
Yes,
Okay.
I also can hear you.
Okay, so good afternoon, everyone. First of all, I would like to thank very much the Symposium for this opportunity. So my name is Rinaldo Rodrigues, and I'm a PhD student in the graduate program in biological sciences at the Federal University of Goiás. And Dr. Fernando Giacchini is my advisor. Today, I'm presenting you a study entitled Reduced O-linked N-acetyl-glucosamine, which we call O-GlcNac, impacts on placental structure and fetal growth in spontaneously hypertensive rats. The hypertensive disorders of pregnancy are characterized by the presence of arterial hypertension during pregnancy with diastolic blood pressure equal to or greater than 90 millimeters of mercury and or systolic blood pressure equal to or greater than 140 millimeters of mercury. It complicates 5 to 10% of all pregnancies, and it is one of the leading causes of maternal and perinatal mortality and morbidity. The hypertensive disorders of pregnancy can be caused by the pregnancy itself, as happens in preeclampsia, or by the presence of chronic hypertension or kidney disease, for example. So, regarding the hypertensive disorders of pregnancy, 4 main types can be highlighted. So, the first one is called chronic hypertension. This is the most common one, and it happens when the woman already presents hypertension before getting pregnant, or when the hypertension appears before the 20th week of pregnancy. The second one is called gestational hypertension. This one is the early onset hypertension that develops after 20 weeks of pregnancy, but without proteinuria. This one can be transitional or permanent. The third one is called superimposed preeclampsia. So this one is the presence of proteinuria and the preeclampsia in a woman who presents chronic hypertension. And the last one is the preeclampsia, which is the most severe one. So preeclampsia present multisystemic commitment with the presence of hypertension after 20 weeks of pregnancy, but with the presence of proteinuria or organ dysfunction, such as kidney and liver dysfunction. But regardless its etiology, what the hypertensive disorders of pregnancy have in common is structural and functional changes in an important organ during pregnancy, which is the placenta. So the placenta is crucial to fetal development once it establishes an interface between the maternal and fetal environment, protecting the embryo and providing the proper nutrients and oxygen supply to the growing fetus. It also participates in the maternal recognition of pregnancy and serves at the production of important hormones during pregnancy. So, because the placenta is crucial during pregnancy, a successful placentation is a key event for a successful and healthy growth of a baby. So during this moment, several signaling pathways are recruited to modulate and ensure these such important processes. So during placentation, many important proteins in the placenta are known to be a target for post-translational modifications with glycans. And here we can highlight the O-glycosylation with a saccharide that is called N-acetyl-glucosamine, which we call O-GlcNac. So O-GlcNac is a dynamic and reversible post-translational modification by the addition of one single saccharide to the serine, threonine, and tyrosine residues of proteins of cytoplasmic and nuclear proteins. And by its actions, O-GlcNac alters protein function and fate, and consequently control cellular process and cellular responses. The most important source for O-GlcNac formation is what is called hexosamine biosynthetic pathway, the HBP. So the glucose enters the cell and it can go to the glycolytic pathway or the HBP. Once glucose goes to the HBP, glucose is converted into glucosamine 6-phosphate by a GFAT enzyme. And subsequently, glucosamine is converted into uridine diphosphate gluconac, which serves as a donor for another enzyme, which is called O-GlcNac transferase, that will be responsible for the addition of the gluconac in the hydroxyl of the serine, threonine, and tyrosine residues of proteins. A third enzyme, which is called O-GlcNacase, or OGA, is responsible for the removal of the O-GlcNac from proteins. The glucose enters the mother's body and reaches the growing fetus through the uteroplacental circulation. In the placenta, the glucose, the uptake of glucose happens through glucose transporters. And the main glucose transporters in the placenta responsible for the glucose uptake are the glucose transporter 1 and glucose transporter 3. Once the glucose enters the placenta, it is metabolized and stored in the form of glycogen cells. Interestingly, during hypertensive disorders of pregnancy, the balance between glucose uptake and glucose transporter, as well as glycogen cells formation, are known to be disrupted. Thus, the hypothesis of the present study is that during hypertensive disorders of pregnancy, alterations in the HBP pathway leads to reduced O-GlcNac in the placenta, which leads to placental structural and functional derangement, which leads to impaired fetal growth and what we call small for gestational age offspring. To achieve this, upon approval by the Committee in Ethics in the Use of Animals, spontaneously hypertensive rats and Wistar rats were separated into hypertensive and normotensive group, respectively. The rats were submitted to mating, and after 20 days of pregnancy, the rats were killed, and the gestational parameters were analyzed, and the placentas were removed to posterior Western blot immunohistochemistry and periodic acid sheaf PAS analysis. The statistical analysis was performed using student t-test, a novel one-way followed by Tukey post-test, and Fisher's exact test. And the values of p lower than 0.05 was considered to be statistically significant. So, first of all, we observed, as expected, that spontaneously hypertensive rats present a higher systolic blood pressure when compared to Wistar rats. This is expected because this animal model naturally presents increase in blood pressure, because these animals are more sensitive to sodium and the angiotensin II actions, which is similar to the essential hypertension that we see in humans. Moreover, when we look to the pregnancy, we saw that the DAMS SHR presented a lower body weight gain when compared to the Wistarette since the very first week of pregnancy. According to the lower body weight gain, we observed that after birth, the litter weight was found to be reduced in SHR animals. And although we do not see any differences in number of the fetuses, we found that SHR presented increased number of pre-implantation loss. So when we look to the fetal weight, we observed that SHR animals presented reduced fetal weight when compared to Wistarettes. And moreover, we observed that 100% of the fetuses was found to be small for gestational age, while none of them was found to be appropriate for gestational age. So when we look to the fetal weight, we need to look to the placenta. And when we look to the placenta in SHR, what we see is that the placenta weight is increased in placentas from SHR when compared to Wistarettes. And while the placental efficiency was found to be reduced. This leads us to believe that the placenta is getting bigger in order to achieve a proper nutritional demand for the fetus, but this placenta isn't functional once we see that the fetus are smaller and the placental efficiency is reduced. So we decided to understand what is going on in the placental tissue. And when we performed morphometric analysis in the placenta, we observed an alteration in two main parts of the placenta, which is the junctional zone and the labyrinth area of the placenta. We observed that SHR presented reduced area of the junctional zone of the placentas, while the labyrinth area was found to be increased. So here we can see the junctional zone of the placenta and the labyrinth area of the placenta, the two main parts of the placenta. Here in the junctional zone, we have glycogen cells and cells that ensure the energetic apport to the growing fetus, and cells that help to produce hormones during pregnancy. And here we can see the labyrinth, where is the area where the exchange between mother and fetus blood happens. So here is the area of blood exchange. So these are the two main parts of the placenta where we observed altered morphometry. Previous work in our work team have already established a link between the O-GlcNac deposition in the placenta and altered placental morphometry in a hyperglycemic animals model. So we aim to understand what is happening with the O-GlcNac in the placenta of spontaneously hypertensive rats. And surprisingly, what we saw is that in SHR animals, we observe a reduced of the placental O-GlcNac expression in these animals, as we can see here in this picture. When we performed immunohistochemistry of the placenta, we observed that there is a reduction of O-GlcNac deposition in the junctional zone of the placenta. Here, highlighted by the black arrows, where we can see the giant cells of the placenta and spongiotrophoblast cells of the placenta. In the labyrinth, we also observe that there is a reduced of O-GlcNac deposition in the placental tissue in trophoblast cells and endothelial cells of the labyrinth region of the placenta from SHR animals. So we decided to understand what is happening in the HBP, that is the pathway that allows the O-GlcNac. So when we looked to the OGT, OGA, and GFAT enzymes, we do not observe any differences between these enzymes in the SHR when compared to Easter rats. Just for us to remember, the defect, the OGT and OGA enzymes work in the HBP pathway to allow the addition of and the removal of the O-GlcNac. So we decided to understand what is happening to the glucose uptake in the placenta from these animals. And when we performed the PAS analysis, we observed that there is a reduction in the number of glycogen cells deposition in the junctional zone of the placentas from SHR animals, as seen here by the black arrows. Here in this picture, we can see again the junctional zone of the placenta with the glycogen cells deposition here. And here we can see that in the SHR, there is a reduced in the placental deposition of glycogen cells. So we decided to understand what is going on with the glucose transporter of the placenta. So we looked to the glucose transporter 1, and what we found was that there was a reduced in the placental glucose transporter 1 expression in the SHR animals when compared to Eastern rats, as seen here by this picture. This could be related to the reduced glycogen cells deposition that we observed and the O-GlcNac reduction that we also observed. Our results are in accordance with the literature that also find a relationship between the gluconac deposition in the placenta and its importance during pregnancy, and also the importance of the glycogen cells and how it is disturbed during hypertensive condition. So, summarizing, what we found here was that during hypertensive pregnancy, we saw reduced in the O-GlcNac in the placentas, mainly in the two main parts of the placenta, which is the junctional zone and the labyrinth, concomitant with alterations in the morphometry of these two important layers of the placenta, which leads to a placental structural and functional derangement, which could be related to the small for gestational age offspring that we observed. So, what is next? We aim to investigate how is glucose transporter 3 in these animals once we only look to the glucose transporter 1, so we can understand better how is happening this decreased nutritional apport to the placenta. And we would like to investigate how the lack of O-GlcNac observed is affecting the placental structure and function. And here we aim to understand which signaling pathway could be involved. And finally, we would like to perform the same analysis early in gestation. So we would like to perform this analysis in 14 and 17 days of pregnancy. So we could observe that the reduced protein O-gluconesylation resulting from the decreased placental nutritional apport contributes to placental structural and functional derangement during hypertensive pregnancy, resulting in fetal growth restriction. I would like to thank the lab where I work, my work team and colleagues, our co-workers, and the source of funding. I also would like to thank very much the Symposium and the program of Biological Sciences for this amazing opportunity. Thank you very much.
No questions for a while. Rinaldo, I have a question. In fact, a simple question. Maybe I lost this information during your presentation. In pregnant women or female rats, for example, with higher blood pressure, does the fetus also have higher blood pressure?
The fetuses?
Yes.
No, we do not perform any analysis in the fetuses, but no, I don't think so. But the offspring have the higher risk to develop, to suffer from these consequences when they grow up. And that's an interesting question because the blood pressure from the SHR animals start raising later, not soon as they are born, actually.
Yes, I see. Thank you, Rinaldo. Mauro, do you have any questions?
Yes, I have one. I was wondering what is the risk for the mother during this pregnancy hypertension? It's the risk. It's for the fetus, the offspring, or the risk will be for the mother during this process.
During hypertensive disorders of pregnancy, the risk is for both. The fetuses can suffer from it, and the babies, and for the mothers too. For example, what happens in preeclampsia is that the mother can evolve to the eclampsia itself, where the mothers can develop seizures. So yes, it presents risk to both mother and fetus, because this could be explained by the fact that the preeclampsia is still one of the most causes of mortality during birth, for example.
And when you look at this structural this placental structure, can you tell if this problem will also affect how the solutes and nutrients pass from the mother to the fetus? I mean, we know that we have a structural alteration, but it's also interesting to know in the microvasculature if there also these differences.
Yes, it would be really interesting to look up for. We do not analyze the impact of the nutritional part in the fetuses, but I think there is an impact of the alteration of this, because this is the placentas from the hypertensive animals, for example, are present a hypoxia environment. So I believe that this is related to the reduce of the nutritional part to the growing babies.
And how about the drug that you are using here? You know if this drug can access the fetus, the offspring during the treatment?
I'm not using drugs in these experiments. I'm just analyzing the O-GlcNac, which is a process that happens. It's a physiological process. So, but we can perform studies with drugs. There is pharmacological tools to increase, for example, the O-GlcNac levels. So we have glucosamine treatment, which is a drug that could increase the global levels of O-GlcNac. And we also can perform in vitro experiments, for example, with Tiamet-G, which increases the global levels of O-GlcNac. So we did not perform yet using a drug or a pharmacological tool, but it would be really interesting. In fact, we are aiming to do that now in the next steps of this project with human placental explants. So we can treat them with a drug that rises the level of O-GlcNac, so we can see the impact of it in the placental tissue.
Interesting. Thank you.
Thank you.
Thank you, Professor Mauro. Rinaldo, unfortunately, we have more questions. We also had some compliments for you, but we have to finish because we have to prepare our next section, our next activity. Thank you again, Rinaldo, for this beautiful presentation. So now we are going to have a little break, and we will be back soon for the next section at 4:15 p.m. Thank you very much for your attention. See you soon.